Hello guys, today we are going to start a new subject, that is medical biology. So, first of all, we are going to discuss about mitosis and meiosis. Basically, how can a cell divide? Okay, so it have basically 5 steps in mitosis. So, first one which we are going to discuss is mitosis. You don't have to draw all of these diagrams. This whole thing is just for your understanding, so don't draw this. In the end I will write a summary, you have to write that summary. Okay, mitosis have 5 phases. The first one is interphase, the second one is prophase, third one is metaphase, okay, the fourth one is anaphase, the fifth one is telophase, and the last step we have 2 cells. Okay, and we can also say it as cytokinesis. Cyto means cytoplasm, kinase means its division. Okay, so now this, what happens in this interface? This interface have many subphases. Okay, so the first one is G1. What happen in this G1? The cellular content duplicate. It means whatever the cell have, the content becomes double. Okay, the size of cells start increases and everything. Okay, now after this G1 we have G node. It's arrest phase, or it is also known as resting phase. What? Resting phase. Okay, so it is not usually present in every cell cycle. Okay, what happens in this phase? See, the cell doubles its content in G1 and then it takes a rest, and that resting phase is G node phase. Okay, after this, if cell want to divide, then there is S phase. And what is this S phase? It is known as synthesis of DNA. What happens in this S phase? There is synthesis of DNA. Okay, or to be very precise, we can use replication of DNA. Okay, and in this S phase also there is centriole duplication. The centriole duplicate, or the centrioles number become 2. Okay, centrioles divide. So in S phase the DNA content become double and centrioles duplicate. Okay, then there is phase G2. In this G2, cell double check duplicated chromosome and errors are corrected. Okay, and one more thing, in this phase tubulin protein synthesis. Tubulin, you will study about this tubulin and this centriole, okay, in organs part. Okay, but just remember here, tubulin protein synthesis. Okay, so interphase have G1 phase, G node phase, S phase and G2 phase. Okay, then after this G2 we have prophase. This prophase will start. So remember one thing from here, that in S phase there is synthesis of DNA. Okay, in this we are going to represent the capital N is for DNA and the small n is for chromosomes. What are chromosomes? They are nothing, just there's a structure that carries your DNA. Okay, that is chromosome, or that have your genetic information. That is chromosome that have your genetic information. Okay, and it's present in the nucleus, as you all know. You can see this is your nucleus. Okay, inside this chromosome. Okay, now remember here, in this S phase there is synthesis of DNA. In this there is 4n, but the chromosome doesn't divide here. Okay guys. Okay, now prophase. In prophase, as I told you, now tubulin protein is start synthesizing. As you can see, tubulin protein is start synthesizing in the cell. Now these chromosomes, the spindle fiber will come and attach in the center of here. This spindle fiber is nothing, but it is a tubulin protein. The spindle fiber is tubulin proteins. Now, as I already told you, it is mitotic spindle, and it arises from where? From centriole. Okay, I want to tell you one point here. It's kind of important, and just remember this thing like here, like capital N is for DNA and small n is for chromosome. Okay, now this mitotic spindle can be damaged by some drugs like colchicine. Okay, this drug can destroy spindle fibers. This drug colchicine, this colchicine is the drug of gout. Gout is a problem you will study in biochemistry. Okay, and commonly we use this drug colchicine to study chromosomes. Okay, so in which stage we can study chromosomes? That is metaphase. Remember, that is metaphase, not prophase. Actually in metaphase, these chromosomes, mitotic spindle or spindle fibers start to pull those chromosomes toward themselves. Okay, so when they will start to pull themselves, so there is one stage arise in which these chromosomes will come in center, and that is known as equatorial plate. What? Equatorial plate. Equatorial plate. And what is this equatorial plate? Chromosomes in center. Chromosomes in center. Okay, and we study chromosomes in metaphase. It is also stage to study chromosomes. Okay, and how we study the chromosomes? We put this colchicine medicine in metaphase of the cell, and these spindle fibers get damaged. Okay, so when these are damaged, these chromosomes will be arranged in center, and because of that we can study them properly, or the scientists. Okay, then what happens in anaphase? In anaphase, these spindle fibers will pull towards themselves the chromosomes. Okay, or they divide. So chromosomes divide here, 4n. Okay, chromosomes divide here, but DNA divides, or DNA synthesis at S phase in interphase. Okay, you should remember the sequence IP MET. How to remember the sequence? There is one mnemonic. You know, there is a very famous movie IP Man. From that you can remember IP MET, M-A-T, IP MET. Okay, IP MET. So when does chromosomes divide? Remember these questions. Chromosomes divide here. Okay, and in telophase what happens? The nuclear envelope start to form again, that nuclear membrane. Okay, so here we will see start forming the nuclear membrane, and in that those molecules we see. Okay, this is chromosomes. Okay, remember this thing very clearly. Okay, in interphase there is G1 phase, G node phase, S phase, G2 phase. Okay, after this G2 we have prophase. In S phase there is synthesis of DNA and centriole duplicate here. Okay, in G2 phase tubulin protein synthesize. Okay, and in this G2 phase also cells double check its duplication, like everything is duplicate correctly. Okay, in cancer cells this G2 phase is absent, that's why there is unnecessary division of cell or pathological division of cells. Okay, now in prophase, as I told you, what is prophase? In this prophase, this spindle, mitotic spindles, these are also known as spindle fibers, will join to chromosomes in the center. Okay, and from there they will pull them, and when they will pull them, then the second phase arises, that is metaphase. Okay, and in this phase we see equatorial plate. This line, when chromosomes are arranged in a line, this is known as equatorial plate. Okay, now in anaphase the chromosome divide here. Okay, chromosomes. In telophase we can see this, and cytokinesis, in this what we see? In cytokinesis we see the cells, the cytoplasm divide properly, and the two cells form. Okay, of 2 small n, 2 small n. Okay, like this is 4n, 4 small n. I hope this thing is clear. Okay, now I want to tell you something about meiosis. See, meiosis is approximately same with mitosis, just there is one difference in meiosis. Like in meiosis there is difference in prophase 1. Okay, this prophase 1 have many stages like leptotene, zygotene, pecitine, diplotene and dikinases. Okay, this liptotin is start to condense. Actually, what happens? See, the first interface is exactly same. Okay, in meiosis also. So as you know that this DNA is divided here. Okay, now this divided DNA is start to condense, or that chromosomes. Obviously, the DNA is inside the chromosomes. Okay, that start to condense. Now in zygote, what happens? The synapse begin. Actually, what happens? See, I want to explain by this diagram. Like this is interface. As you can see that it's divided. Okay, this thing. The DNA content divided, but chromosome is 1. Okay, then in prophase 1, what happens? As you can see, these start to condense. These start to condense, and when they will condense, they will form an X-like structure like this. X and X. Okay, then this is their condensed stage. They condensed like this. Now in zygote, synapse begin. Synapse begins, and sometimes it's also called like bivalent forming. Actually, what happened? Now these two chromosomes will transfer their data, some data to one and some data to another. Okay, how they will come close to each other like this and overlap. Okay, so this is synapse begin. They come and overlap each other. Actually, what they are doing in this overlapping, they are transferring or sharing their information, or they are changing their information from one to another. Okay, now pectine. In this pectine, synapse complete, or it is also known as crossing over occurs. Okay, so they are coming close to each other. Okay, now in this synapse complete, what happens? Whatever they are sharing the information with each other. Okay, the one is giving, like this is 1, this is 2. The one is giving some information to 2, and 2 is giving some information to 1. Okay, so in this stage, what happens? They done whatever they want to do, like they transfer their information, and then the next stage comes, that is diplotin. And what happens in diplotin? We see chiasma. Chiasma is visible. Chiasma is visible. What is chiasma? Actually, that information that both of them shared with each other, that we can see. And how we can see? Let me show you by a little diagram. Okay. Okay. So, like this. This green one is the information of first part. Okay. So we see this is chiasma. Chiasma is visible. You can see this. As I told you, this is first one, this is second one. This is first one, this is second one. So some information of first, this first one is given to second, and some information of second is given to first, and we can see this. Okay, you can see this box. Okay, this is chiasma. Now we can see chiasma is visible. And in dikinases, what happens? Now these two cells go for metaphase 1. Now go for metaphase first. Okay, now they go for metaphase first. Okay, and then the cycles is same, like they start to divide. Okay, after metaphase they divide into 2 cells. Okay, and after division, then second divisions happen in meiosis. This is meiosis 1, from here to here. Okay, this is meiosis 1, and from here to here it's meiosis 2. Actually, what happens in meiosis 2? See, this cell divide into 2. Okay, it's same as mitosis. Okay, the just difference is in this prophase 1, and we just discuss about it. After this, then these cells again divide. Okay, and they make haploid cells. Haploid means, see, if they have 2n, then they will have small n of chromosomes. Okay, this thing is clear. See, here we have 2n, then it became again 2n, like same as mitosis, but this 2n will become n. Okay, so now, as I told you, I hope you understand this thing. Okay, I told you don't have to write this. Now I am going to write a summary part, and that you have to write, or you must have to write. So let's start that summary part. First of all, as I told you, in interface, G node is resting phase. G1 is cellular content duplicate here. Okay, then there is S phase, and this S is also known as synthesis phase. Okay, in this what happened, there is synthesis of DNA that you must have to write. Okay, but chromosome is same, but chromosome number is same. Okay, and in this S also there is one more thing, that is centriole duplicate. What is centriole? These structures from which these tubulin proteins form, these are centrioles. Okay, and they will form this spindle fibers that will go and attach in the center of this chromosome. Okay, centriole duplicate. Now there is G2, cell double check and correct the error. And one more thing, tubulin protein synthesized. Okay, here tubulin protein synthesized. Now between mitosis and meiosis. Mitosis, as I already told you about interphase, this is like interphase and it's common in both the cells. Okay, and after this you can also say, like, after interphase there is two, like mitotic division or meiosis. Meiosis is also known as reductional, as I already told you. Reductional. Why reductional? Reductional division. Why? Because in this the haploid is forming. Okay, now this mitosis and meiosis. Okay, now after interphase, what is the phase? That is mitosis and meiosis. Now in mitosis we have prophase. In this prophase, nucleoli disappear. What is this nucleoli? This outer membrane of nucleus. Okay, as you can see, here is this membrane, but here we have no membrane in prophase. Okay, so this nuclear membrane or nucleoli disappear in prophase. Okay, nucleoli disappear. In this, chromatin is formed, and in this spindle fibers form. Okay, and one more thing happens here, that centriole goes to opposite pole. Centriole goes to opposite pole. Pole means, see, this is your cell, na? This have one and this is like second pole. So centrioles move to poles. Okay, after this, if these spindle fibers, I saw one question, if they are not formed, then chromosome won't go to other poles. Okay, if not formed, then chromosome won't go to other poles if this spindle fiber won't form. Okay, now the second one is after prophase is metaphase. This metaphase, as I already told you, we see equatorial plate. Okay, and it is for best studying phase. Okay, in this usually all of the scientists study the cell divisions or about chromosomes. Why? Because in this there is maximum amount of condensed chromosome. Okay, and as I already told you, for studying what chemical we put in the cell, that is colchicine. Okay, now after metaphase, which phase we have in mitosis? That is anaphase. What? Anaphase. What happens in anaphase? The first one is 4n. 4n means chromosome move or go to opposite pole. Basically, in this stage, how many, for a human, we have 92 chromosome. Normally, how many chromosomes we have? We have 46. Okay, but in this stage, we have 92. Okay, now there is telophase after this. Two cells formed. We can see the two cells formed properly in just an 8-like structure. 8, 1, 2, 3, 8, that 8-like structure. Okay, but the cytoplasm doesn't divide. Okay, so two cells formed, and then we have cytokinesis. What is cytokinesis? Two different daughter cells form. Two different cells separate, and we have 2n and 2n, and both have 46 and 46 number of chromosomes. Okay, this thing is clear for mitosis. Now come to meiosis. This meiosis, as I already told you, it only have difference in its prophase 1, and all the questions come from this prophase 1 part. Okay, it have leptotene, zygotine, pectine, diplotene, dikinesis. Okay, lapet do, jod do. Okay, now what is leptotene? Chromosome start condensing. Okay, in zygotine we see synapse begins or bivalent form. Pectine is synapse complete. Okay, or we can also say it as crossing over occurs. Now this diplotene is, in this what we see, chiasma is visible. Chiasma visible. Now this dikinesis is bivalent, that cell go for metaphase 1. Metaphase 1.